Hi friends, welcome back to my another video tutorial. So in this section, I'm going to explain about the transport of respiratory gases. First of all, the transport of oxygen transport. See, we know respiration. The concept of respiration is simply energy production. Energy production takes place in our cell, and our body utilizes atmospheric oxygen for the production of energy within the cell. By a process of inhalation, atmospheric oxygen enters the lung alveoli. And from the lung alveoli, this oxygen diffuses to the blood. And through the blood, this oxygen transported to the capillary surrounding tissue. And from that capillary surrounding tissue, this oxygen diffuses to the cell. And as a result of oxygen utilization within the cell, the carbon dioxide produced in the cell. That produced carbon dioxide diffuses to the capillary surrounding blood, and through the blood this carbon dioxide transported to the lung alveoli and eliminated by a process of expiration. So here the transport of respiratory gases means transport of oxygen from lungs to systemic tissue. Similarly, transport of carbon dioxide from systemic tissues to lungs. This form the respiratory gas transport. So oxygen transport means by the inhalation, oxygen reaches the lung alveoli. And from this lung alveoli, oxygen transported to the capillary surrounding this tissue. So oxygen transported from alveoli to systemic tissues. This transport of oxygen from alveoli to tissues in two forms. First one, in dissolved state. First form in dissolved state. Second form as oxyhemoglobin. Oxyhemoglobin. So oxygen transported from lungs to tissues in two forms. They are, one is in dissolved state, another one is in chemical combination with the hemoglobin called as oxyhemoglobin form. This one is in dissolved state. 3% of oxygen. Only 3% of oxygen transported in dissolved state. 3% of oxygen transported in dissolved state through the plasma water. Percentage of oxygen transported in dissolved state. One is through plasma water, water in plasma, as well as water in blood cells. In dissolved state through the plasma water as well as the water in blood cell. 97% of oxygen, almost all oxygen, 97% of oxygen transported by the hemoglobin. 97% of oxygen transported by the hemoglobin in RBC. 97% of oxygen transported by hemoglobin in RBC means in chemical combination with the hemoglobin, oxygen transported in the form of chemical combination with the hemoglobin called as oxyhemoglobin form. Now, hemoglobin is actually a conjugated protein. This hemoglobin is a conjugated protein, means it has a protein part as well as a non-protein part. So it has protein and non-protein part. The protein part of hemoglobin is called as globin. Non-protein part of hemoglobin is called heme. This protein part or globin, actually 4 globulins or 4 polypeptide in a hemoglobin. So the protein part or globin means total 4 globulins or 4 polypeptide in a single hemoglobin. They are 2 alpha chains, alpha 1 and alpha 2 chains, 2 alpha chains. Similarly 2 beta chains, beta 1 and beta 2 chains, 2 alpha chains and 2 beta chains. This is the protein part of hemoglobin, globin. Total 4 globulins, they are alpha chain, 2 alpha polypeptide chain and 2 beta polypeptide chain. Then the non-protein part of hemoglobin is heme. This heme is formed of protoporphyrin 9, protoporphyrin 9 and iron. this iron in the form of ferrous state. This is the non-protein part. This non-protein part is heme formed of porphyrin ion and iron in the form of ferrous state. And here this protoporphyrin ion. So this protoporphyrin ion is formed of the amino acid glycine combined with the succinyl coenzyme A. Succinyl coenzyme A is a product formed in Krebs cycle. So the succinyl coenzyme of Krebs cycle combined with the amino acid glycine to form a pyrrole ring. A pyrrole ring. Suppose this is a pyrrole ring. And 4 pyrrole ring combine to form a protoporphyrin. 4 pyrrole ring combine to form a protoporphyrin. This 4 pyrrole combine to form a protoporphyrin. And this protoporphyrin with a ferrous ion to form a ferrous ion and in the form of ferrous state to form the non-protein part is called as heme. So this is the heme, the non-protein part of hemoglobin. Each polypeptide combined with one heme group. This heme group combined with each polypeptide. That means a single hemoglobin has 4 globin chains as well as 4 heme. See, this iron in the heme can carry oxygen. This iron in the heme can carry oxygen. So in hemoglobin, oxygen is transported by the iron. One heme can carry 1 molecule of oxygen. Thus a single hemoglobin can carry a maximum of 4 molecules of oxygen, because a single hemoglobin consisting 4 heme and each heme can carry 1 molecule of oxygen. Therefore a single hemoglobin can carry a maximum of 4 molecules of oxygen. This is the fully saturated form of hemoglobin with oxygen. When 4 molecules of oxygen combined with the hemoglobin is known as the fully saturated form of hemoglobin. But we know the same hemoglobin can carry carbon dioxide. It can bind with the H plus ion because this hemoglobin is a buffer. So in a hemoglobin acts as a buffer. Because the histidine amino acid of this hemoglobin can combine with the H plus. The histidine amino acid of this hemoglobin can combine with the H plus ion. Thereby it acts as a buffer. It combines with the H plus ion. Carbon dioxide is transported by the amino end of this polypeptide. That means this carbon dioxide is transported by this protein part. Histidine also combined with the protein part of hemoglobin because the histidine amino acid in this globin can combine with H plus ion. So that means this hemoglobin not only carry oxygen, it can carry carbon dioxide, then it can carry H plus ion, then it can carry carbon monoxide also. This hemoglobin can carry carbon monoxide. But this carbon monoxide binds to the iron of this hemoglobin. The same iron in the hemoglobin can carry carbon monoxide. That means there is a competition between oxygen and carbon monoxide for hemoglobin, because both oxygen and carbon monoxide are transported by the iron in the hemoglobin. But the hemoglobin, this hemoglobin has about 200 to 300 times more affinity to this carbon monoxide than oxygen. Hemoglobin has about 200 times more affinity to carbon monoxide than oxygen. See, 100 ml of blood. 100 ml of blood contains on an average of 15 gram of hemoglobin. 100 ml of blood contains on an average of 15 gram of hemoglobin. And here, 1 gram of hemoglobin can carry 1.34 ml of oxygen. 1 gram of hemoglobin can carry 1.34 ml of oxygen. Therefore, 100 ml of blood can carry. Therefore, 100 ml of blood can carry 15 into 1.34 ml of oxygen. 100 ml of blood can carry 15 into 1.34 ml of oxygen because 100 ml of blood contain on an average 15 gram of hemoglobin. And among this 15 gram, 1 gram can carry 1.34 ml of oxygen. Thus, therefore, a 100 ml of blood can carry 15 into 1.34 ml of oxygen. Approximately 20 ml of oxygen. So 100 ml of blood can carry approximately 20 ml of oxygen. Approximately 20 ml of oxygen to the tissues. Suppose this form tissue and this is 100 ml of blood, and this 100 ml of blood can carry 20 ml of oxygen to the tissues. So 100 ml of blood can carry 20 ml of oxygen to the tissue for the metabolism or for the energy production. But under the normal physiological condition, our body required only lesser amount of energy under the normal physiological condition. So for the normal physiological activity, body required only lesser amount of energy. That means body required only lesser amount of oxygen for the production of energy. So for the normal physiological activity, every 100 ml of blood delivers only 5 ml of oxygen to the tissues. See, this 100 ml of blood can carry 20 ml of oxygen, but for the normal physiological activity, this 100 ml of blood delivers only 5 ml of oxygen to the tissue for the normal physiological activity. So only 25% of oxygen delivers to the tissue for the normal physiological activity. The remaining 15 ml, that means 75% of oxygen left unused in blood. 75% of oxygen left unused in blood. And this 75% of oxygen or this 15 ml of oxygen in this blood actually used as the reserve for the active metabolic process or for a active muscular exercise. Body use this reserve. So this remaining 15 ml is actually a reserve for the active metabolic process, and this is why only 5 ml of oxygen delivers to the normal physiological activity. And when our body required large amount of energy for an active metabolic process, this remaining blood acts as a reserve, and this reserve utilized for that active muscular process. For the transport of oxygen, hemoglobin combine with the oxygen and form oxyhemoglobin. This process is known as oxygenation. So this occurs near the alveoli and near the tissue. Near the tissue, this oxyhemoglobin dissociate to form hemoglobin and oxygen for the utilization of oxygen in tissue. This is deoxygenation. This process is deoxygenation. So oxygenation and deoxygenation, because hemoglobin reversibly bind with the oxygen to form oxyhemoglobin. This oxyhemoglobin is not stable. It associated and dissociated in different part of the body like alveoli and tissue. That means both oxygenation and deoxygenation takes place in our body. This oxygenation and deoxygenation, that means the association of hemoglobin with oxygen and dissociation of hemoglobin from oxygen, depends on some factors. Then what are the factors affect the association and dissociation of hemoglobin and oxygen? So factors affecting Association and dissociation of hemoglobin and oxygen. That factors includes PO2, partial pressure of oxygen. Partial pressure of oxygen is the most important factor that affect the association and dissociation of hemoglobin and oxygen. So first factor is partial pressure of oxygen. This is the most important factor because when PO2 increases, the binding of hemoglobin with oxygen also increases. That means the percentage of saturation of hemoglobin with oxygen also increases when partial pressure of oxygen increases. When this partial pressure decreases, the affinity of hemoglobin to the oxygen will also decreases. So the oxyhemoglobin dissociation will occur. So this PO2 is one of the most important factors which affect the association and dissociation of hemoglobin and oxygen. Another factor is partial pressure of carbon dioxide. This partial pressure of carbon dioxide is another factor which affects the association and dissociation of hemoglobin and oxygen. Because we know in hemoglobin, oxygen is transported by the ion in the hemoglobin. But carbon dioxide is transported by the amino end or the protein part of this hemoglobin. That means there is no competition between oxygen and carbon dioxide for hemoglobin. Because this oxygen and carbon dioxide are transported by different site of hemoglobin. Oxygen is transported by ion, carbon dioxide is transported by the protein part of hemoglobin. But the association of hemoglobin with oxygen, similarly the association of hemoglobin with carbon dioxide is not a strong covalent bond. It is a weak association. It is a very unstable formation. So in this oxyhemoglobin, oxygen is not strongly attached to this hemoglobin. It's not a strong covalent bond. But this oxygen very loosely bounded to this hemoglobin. And we know carbon dioxide transported by the protein part of this hemoglobin. Suppose in this oxyhemoglobin, when carbon dioxide binded to the protein part of this hemoglobin or the amino end of this hemoglobin, there is a conformational change occurs in hemoglobin. As a result, the bond between oxygen and hemoglobin broken, which causes the unloading of oxygen from this hemoglobin. When carbon dioxide binded to the amino end of hemoglobin, the bond between oxygen and hemoglobin broken, which causes the unloading of oxygen from this hemoglobin. So there is no competition between oxygen and carbon dioxide for hemoglobin because they bind on entirely different side. But when this carbon dioxide bind of hemoglobin, this bond between oxygen and hemoglobin broken causes the unloading of oxygen from hemoglobin. Similarly, when oxygen bound on this hemoglobin, what happened? This carbon dioxide is unloaded. That means the conformational change occurs in hemoglobin causes the unloading of carbon dioxide from this hemoglobin. So the partial pressure of carbon dioxide also affect the association of hemoglobin and oxygen because when the PCO2 is higher, carbon dioxide bind on this hemoglobin causes the unloading of oxygen from hemoglobin. We know near the capillary surrounding tissue, the partial pressure of carbon dioxide is very higher. At this high PCO2, this carbon dioxide bind on the amino end of hemoglobin causes the unloading of oxygen from hemoglobin. This is why carbon dioxide is a factor which affects the association and dissociation of hemoglobin and oxygen. Then third factor is hydrogen ion concentration. H plus ion or pH. pH is another factor which affects the association and dissociation of hemoglobin and oxygen. Like carbon dioxide. Suppose when oxygen in combination with the hemoglobin in this oxyhemoglobin form, when H plus binded to the histidine amino acid in this hemoglobin, this histidine is the buffer in this hemoglobin. When H plus bind on this histidine amino acid in hemoglobin, what happens? There is a slight conformational change in hemoglobin causes the unloading of oxygen from this hemoglobin. When H plus bind on the histidine amino acid of this hemoglobin, what happened? The bond between oxygen and hemoglobin broken causes the unloading of oxygen from this hemoglobin, that is oxyhemoglobin dissociation will occur. When the H plus ion concentration increases, what happened? The dissociation of hemoglobin from the oxygen takes place. So pH is a factor which affects the association and dissociation of hemoglobin and oxygen. Another factor is 2,3-bisphosphoglycerate. 2,3-bisphosphoglycerate. Hemoglobin has affinity to this 2,3-bisphosphoglycerate. This 2,3-bisphosphoglycerate binded to the amino end of beta chains. This 2,3-bisphosphoglycerate binded to the amino end of beta chains of hemoglobin. We know hemoglobin has 2 alpha and 2 beta chains. And this 2,3-bisphosphoglycerate binded to the amino end of these beta chains. And when this 2,3-bisphosphoglycerate binded to the amino end of this beta chain, what happened? The conformational change occurs in hemoglobin, causes the unloading of this oxygen from hemoglobin. So this 2,3-biphosphoglycerate is another factor which affects the association and dissociation of hemoglobin and oxygen. So when this 2,3-bisphosphoglycerate concentration increases, it binds to the hemoglobin, causes the unloading of oxygen from this hemoglobin. But in our fetal life, this fetal hemoglobin is structurally different from adult hemoglobin. Fetal hemoglobin is structurally different from adult hemoglobin. In fetal hemoglobin also have protein as well as non-protein part. The protein part is globin, total 4 globins. The globulins in fetal hemoglobins are 2 alpha and 2 gamma chains. In fetal hemoglobin, the globulin or the protein part include 2 alpha chains and 2 gamma chains. 2 alpha and 2 gamma chain, no beta chains. No beta chains in fetal hemoglobin. That means this fetal hemoglobin has no affinity to this 2,3-bisphosphoglycerate. Fetal hemoglobin has no affinity to this BPG. As a result, this fetal hemoglobin has about 30% more affinity to oxygen. This fetal hemoglobin has about 30% more affinity to oxygen than adult hemoglobin. This fetal hemoglobin has about 30% of more affinity to oxygen than the adult hemoglobin. So by the time when the blood reaches placenta, there is a lower concentration of oxygen in mother's blood. By the time when blood reaches the placenta, there is lower concentration of oxygen in mother's blood. Since the fetal hemoglobin has a higher affinity to oxygen, even in the low concentration of oxygen, hemoglobin in the fetal hemoglobin can carry oxygen to the fetal blood circulation or fetal body tissue. So fetal hemoglobin has higher affinity to oxygen than adult hemoglobin. Even in the low concentration of oxygen in the maternal blood in placenta, this fetal hemoglobin can carry oxygen to the fetal tissue because it has more affinity to oxygen than the adult hemoglobin. So another factor is temperature. Temperature means heat is a byproduct of metabolism. So the region having higher metabolism means that heat is higher or temperature is higher. That means near the tissue, temperature is higher because these are the site of metabolism. As a result of metabolism, large amount of heat produced. As a result of heat, this temperature increases. So this heat is produced as a result of metabolism. That means these tissues are metabolically active region. And for the metabolism, this region required large amount of oxygen. For the metabolism, this region required large amount of oxygen. So the region having high temperature means that region required large amount of oxygen for the metabolism. So the high temperature means that region required large amount of oxygen. As a result, the oxyhemoglobin dissociation will occur. Oxygen dissociated from the hemoglobin. So these are some factors affect the association and dissociation of hemoglobin and oxygen. The factors are partial pressure of oxygen, partial pressure of carbon dioxide, H plus ion concentration or body pH, then 2,3-bisphosphoglycerate concentration and temperature. Then the conditions favorable for the association of hemoglobin and oxygen. The conditions favorable for association of hemoglobin and oxygen includes high PO2. At high PO2, more hemoglobin get binded to the oxygen and oxyhemoglobin association will occur. Then another factor is low PCO2, low pressure of carbon dioxide. Then low H plus ion concentration. Low H plus ion concentration means H plus ion means the hydrogen ion concentration represents the pH. Low H plus ion concentration means the pH is high pH. Low H plus ion concentration means the pH is high, the pH is more than 7. So low H plus ion concentration or in high pH, the association will occur, hemoglobin and oxygen association will occur. So another one is 2,3-biphosphoglycerate concentration. When this 2,3-biphosphoglycerate concentration decreases, the association of hemoglobin and oxygen will occur. Then the temperature. Means low temperature is favorable for the association of hemoglobin and oxygen. Low temperature means that medium required lesser amount of oxygen for the metabolism because that region is not metabolically active. That oxygen required lesser amount of oxygen. That means there is the dissociation rate is comparatively lesser. So these are the conditions favorable for the association of hemoglobin and oxygen: high PO2, low PCO2, low H plus ion, low concentration of 2,3-bisphosphoglycerate and low temperature. Then condition favorable for the dissociation of hemoglobin and oxygen. The conditions favorable for the dissociation of hemoglobin and oxygen. The conditions are low PO2. At low PO2, hemoglobin dissociated from the oxygen. So low PO2. Then high PCO2. We know when carbon dioxide concentration increases, when this carbon dioxide binded to hemoglobin, what happened when this carbon dioxide binded to the amino end of this hemoglobin? The bond between oxygen and hemoglobin broken causes the unloading of oxygen from hemoglobin. So another factor, PCO2. High PCO2 is favorable for dissociation. Similarly, high H plus ion concentration. High H plus ion concentration means that medium is acidic. High H plus ion concentration means low pH. That medium is acidic medium. So in this acidic medium or in this high H plus ion concentration, what happened? Hemoglobin dissociated from the oxygen. Because when H plus ion also binded to hemoglobin, the unloading of oxygen from the hemoglobin takes place. Then high concentration of 2,3-bisphosphoglycerate is another condition favorable for the dissociation of hemoglobin from oxygen, as well as high temperature. These are the favorable conditions for dissociation of hemoglobin and oxygen. Next is oxygen dissociation curve or oxyhemoglobin dissociation curve. This curve means the relation between percentage of saturation of hemoglobin with oxygen. The relation between percentage of saturation of hemoglobin with oxygen and the partial pressure of oxygen. The relation between the percentage of saturation of hemoglobin with oxygen and partial pressure of oxygen represented in the form of a S-shaped curve or in the form of a sigmoid curve. In the form of a sigmoid curve or S-shaped curve called as oxygen dissociation curve or oxyhemoglobin dissociation curve. See, in this curve, when the partial pressure of oxygen increases, what happened to the percentage of saturation? The percentage of saturation of hemoglobin with oxygen will also increases. When PO2 increases, the percentage of saturation also increases. But this oxygen dissociation curve represents the association of hemoglobin with oxygen under the normal physiological condition. The association and dissociation of hemoglobin with oxygen under the normal physiological condition. See, in our body, under the normal physiological condition, not in an active muscular exercise, under the normal physiological condition, when the PO2 at 25 mmHg, 50% of hemoglobin are saturated with oxygen. See, and in this 25 mmHg, 50% of hemoglobin are saturated with oxygen. 25 mmHg means these are the deoxygenated blood. The deoxygenated blood in contracting skeletal muscles. In the deoxygenated blood of contracting skeletal muscle where the PO2 is only 25 mmHg, at this 25 mmHg, 50% of hemoglobin are saturated with oxygen. This is the P50 value under the normal physiological condition. P50 value means the partial pressure of oxygen. The partial pressure of oxygen in which 50% of hemoglobin are saturated with the oxygen called as P50 value. So in the normal physiological condition, our P50 value is normally 25 mmHg in an average healthy individual. So what is P50 value? The partial pressure of oxygen in which 50% of hemoglobin are saturated with oxygen. So our P50 value is under the normal physiological condition is 25 mmHg. Okay. Then when the PO2 increases to about 40 mmHg, about 75% of hemoglobin are in saturation with oxygen. See, at 40 mmHg, we know in our systemic veins, that means the deoxygenated blood. The deoxygenated blood in systemic veins and in systemic tissues. We know the partial pressure of oxygen in deoxygenated blood as well as systemic tissue. In deoxygenated blood as well as in systemic tissue, the PO2 is 40 mmHg. We already studied about the partial pressure of oxygen in our systemic tissue. So here at this 40 mmHg, 75% of hemoglobin are saturated with oxygen means in our systemic tissue as well as in deoxygenated blood, about 75% of hemoglobin are saturated with oxygen. That means we know under the normal physiological condition, every 100 ml of blood delivers only 25% of oxygen to the tissue. That means only 100 ml of blood can carry 20 ml of oxygen. Among the 20 ml, only 5 ml of oxygen delivers to the tissue. That means 75% of hemoglobin are saturated with oxygen in the systemic tissue also. This curve represents the oxygen dissociation for normal physiological condition. In normal physiological condition, the systemic tissue record only lesser amount of oxygen. As a result, almost oxygen are saturated with the hemoglobin in this systemic tissue also. Then, at 100 PO2, when the PO2 increases to 100, maximum saturation, 100% saturation. So this 100% saturation in oxygenated blood, 100% saturation, oxygenated blood and near the alveoli. And near the alveoli, 100% saturation because in alveoli as well as oxygenated blood where the PO2 is 95 and above. So in this condition, maximum saturation will occur. So this is the oxygen hemoglobin dissociation curve. This curve represents when the partial pressure of oxygen increases, the percentage of saturation of hemoglobin with oxygen also increases. Then several factors affect this oxygen hemoglobin dissociation curve. We know about the factors, that factors like partial pressure of oxygen, partial pressure of carbon dioxide, H plus ion concentration, 2,3 biphosphoglycerate, then temperature, etc. are the factors affect this oxygen dissociation curve. Next is Bohr effect. Bohr effect. Suppose in a condition hemoglobin are in 100% of saturation with oxygen. In this condition, the H plus ion concentration increases. See, hemoglobin is a good buffer too. When this H plus ion bounded to the histidine amino acid of this hemoglobin, there is a conformational change occur in hemoglobin. As a result, this oxygen is unloaded from this hemoglobin. When H plus ion bounded to this hemoglobin, the bond between hemoglobin and oxygen broken. As a result, the oxygen unloaded from the hemoglobin. So in our body, if in certain condition, when this H plus ion concentration increases, this oxyhemoglobin dissociation takes place. What happened to the percentage saturation when H plus bounded, oxygen unloaded from hemoglobin? As a result, this oxygen hemoglobin dissociation decreases from 100 to 80. So when this H plus concentration increases, oxygen dissociation will occur. Similarly, when carbon dioxide concentration increases, when this carbon dioxide bounded to the amino end of hemoglobin, similarly, what happened? When this carbon dioxide bounded, the bond between oxygen and hemoglobin broken, this oxygen unloaded from this hemoglobin. So similarly, carbon dioxide binded to the hemoglobin, what happened? The oxyhemoglobin dissociation will occur. The percentage of saturation of hemoglobin with oxygen also decreases. So in a medium, in certain condition, when carbon dioxide or H plus ion concentration increases, when this H plus or carbon dioxide bounded to hemoglobin, oxygen is unloaded from hemoglobin. So the percentage of saturation decreases. See in this graph, when this carbon dioxide concentration increases or H plus concentration increases, oxygen is unloaded from hemoglobin. So what happened to the percentage of saturation to hemoglobin with oxygen? The percentage of saturation decreases from 100, it decreases to 80. That means this oxyhemoglobin dissociation curve shifted to right. Again, when H plus or carbon dioxide increases, this oxyhemoglobin dissociation curve again shifted to right. That means the percentage of saturation decreases. So, when the H plus or carbon dioxide concentration increases, this oxyhemoglobin dissociation curve shifted to right. That means the oxygen carrying capacity of hemoglobin decreases. This is Bohr effect. So Bohr effect means when H plus or carbon dioxide concentration increases, the oxygen hemoglobin dissociation curve shifted to right and oxygen carrying capacity of hemoglobin decreases, called as Bohr effect. And it is postulated by Christian Bohr in 1904, called as Bohr effect. Near the alveoli, where the partial pressure of oxygen is higher, in this high PO2, hemoglobin combine with oxygen and form oxyhemoglobin. In the form of oxyhemoglobin, this oxygen transported to the tissue, and near the systemic tissue, the PO2 is lesser. In this low PO2, this oxyhemoglobin is not stable. So this oxyhemoglobin dissociated to form hemoglobin and oxygen. And this released oxygen is utilized in the cell for the metabolism. This form the transport of oxygen from lungs to tissue. So in this session, we completed oxygen transport from lungs to tissue. Thank you for watching my videos. Please subscribe, like and share my videos. Okay, see you.